So this last part is going to be more biology. It will eventually get back to the mixing sort of stuff, but it's going to take us a little while to get there. And a lot of this stuff I'm expecting you to be able to answer on an exam too, because it's going to be stuff we've covered in lecture as well. Hopefully you already are familiar with this. First question is basically, why are there chromosomes? Is that chromosomes are a whole bunch of genes linked together, a whole bunch of different codes, Oh, the computer didn't like that. And what do you do with them? Well, you make stuff with them. Okay. What else do you do with them? Well, in reproduction, you got to make copies of all the codes and then make sure that your copies go where they need to go. If you're making 2 new cells, then you got to get a complete set of copies into one cell, complete set of copies into the other cell. You don't want to lose any pieces. Think of how many genes there are. This is just the genes from one of the medium-sized chromosomes that we have, and this isn't even really all the genes on it. There's thousands and thousands and thousands and thousands of codes that we use. And if they were all separate pieces of DNA, and we had to make copies of them, and then make sure the copies got into new cells when we made new cells, we would never do it right. What a chromosome does, the reason why we have chromosomes, is it takes the number of pieces of DNA that you need to be able to copy and move around and gives you a usable, practical number where you're not going to screw it up that often. Humans, we have 46 chromosomes. That actually is a high number, and we do screw it up. Is that it's too many or too few chromosomes going where they're supposed to is actually not that unusual. Happened to me as a fetus. If you know anybody with Down syndrome, it happened to them in the production of a sperm or an egg cell, where they didn't just get an extra chromosome, they got a particular extra chromosome. We'll talk about that in a few minutes. But what a chromosome does is it allows you to reduce the number of pieces you have to deal with when you're making new cells to a manageable number. So you don't make quite that many mistakes. Now, we already talked about how for all of our genes, you get pairs of them. You get a set of genes from mom and a set of genes from dad. Well, you get a set of chromosomes from mom and dad too. And the chromosomes are what are called homologous chromosomes, because you have 2 number 9s. Every one of the number 9s has these same genes on it. May not be exactly the same codes, may not be the same alleles, but they're genes for the same kind of protein. So you've got 2 of each of these genes, even if they don't completely match. Like a pea plant has a gene for height, and if you've got the dominant recessive, you've got 1 dominant code and you got 1 recessive code. Well, it's not. 1 code for the working protein and 1 working for the non-working protein. Anyway, so homologous chromosomes are matched gene for gene. We just answered the first 2 questions on question 1. This is a very old representation of chromosomes for humans. And your next question is, how are they different? They carry different genes, for one, but we're going to talk about how they're physically different. And these are smaller as you go. So some chromosomes are really big, carry lots and lots and lots of genes. Some of them are very small and carry still lots and lots, but nowhere near as many genes. Is that even a little chromosome has lots of genes on it. And one of the things that you can see on these, if I can find my little pointer, each of these has a little narrow spot that's cross-hatched. There, 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 there. It's in different places. We'll do this later in the semester, but when you make copies, you make copies of your chromosomes. You wind up with taking the chromosome apart, making a copy with each one, and they stay connected at those spots. They're called centromeres. And when you see pictures of chromosomes, it pretty much always is chromosomes in a cell that's dividing. Because in a cell that's active, the chromosomes are all spun out because you're using them, you're getting at the codes. To move them around, you want them kind of all, if you know anything about knitting, you have them all skeined up. You want them all wrapped up really tight so that they don't tangle in each other as you move them apart. And so you can see them. And this is why you typically see a chromosome and it kind of looks like this. It may be fatter than this, but it's a copy connected to a copy. And sometimes they are fatter. Because the way they wrap up varies from place to place, from chromosome to chromosome. When they're actually all wrapped up, some of them are long and skinny, some of them have fat spots and thin spots. A lot of them have patterns on them, what they call banding patterns. So somebody who really knows what they're looking at could look at a spread of chromosomes and very quickly go, oh, these are human chromosomes. I recognize, number 5 and number 8 and, number 15. And that means that everybody has not just different genes on them, but we package them differently. We connect them in different places. Some of them are big and some of them are small. Some of them are fat and some of them are thin. Some of them have stripes on them and some of them don't. And that's kind of your question. How do chromosomes differ? They differ a bunch of different ways. Some of them important from a functional aspect, different genes. Some of them more important from an appearance aspect when the cells are dividing and you can see them like this. Something we talked about in the chapter about genetics, but we're going to talk about here, is linkage. Here's chromosome number 11. On chromosome number 11 is a gene for part of hemoglobin. There's a gene for insulin. There's a gene for parathyroid hormone. There's a gene for an enzyme in your liver that helps you do stuff with certain toxins. There's a gene for follicle stimulating hormone. Remember I talked about albinism, being albinos? There's the processor, the color processor, that if it isn't working right, produces albinism. But there's a whole bunch of genes all stuck on the same chromosome. They're linked together. And when genes are linked together, if you have two alleles on the same chromosome, when you pass that chromosome, those two alleles go together. So for instance, if the ABO blood group and the RH group are on the same chromosome, they are not, but if they were, and these were your alleles. You have an A and a negative, and an O and a positive. Well, because they're physically connected to one another, if you pass the A on, the negative's going with it. It's connected. And so that changes the patterns of inheritance. You can't just say, well, they just randomly split up. These are all the different possibilities of mixes. No, because they're physically connected to each other. They are linked. Really screwed people up because none of Mendel's genes were linked. So he didn't think that happened. He didn't think there was any connection between the genes he came up with the law of independent assortment, which meant they all split up independent of one another when you sent them on for all of the different P traits that he studied. And then pretty quickly people found out, oh no, you get packages. Human packages, hair color and eye color. One of the major hair color genes and the major eye color gene are on the same chromosome. So you tend to get populations where it isn't just a hair color and an eye color, it's the two of them together, like blonde hair and blue eyes, or red hair and green eyes. And those genes are linked. So that's linkage. I got my questions over here. On to question 4. How do we use this? Well, as people have been studying genetics, it's been, one of the questions is, where on these chromosomes are the genes? If we can find the location, we maybe can figure out the code, and from the code maybe figure out what the proteins are, all sorts of things that if you can find the location of a gene that does something, how do you do that? I'll give you an example. is that in the 1970s, when people had finally decided that cancer was not just genetic or external or environmental, they realized it was all three. But they knew that it was associated with changes in genes. And then the question was, so what genes are changing? Where are they? Can we track them down and figure out what they're supposed to, those proteins are supposed to be doing? Well, they said, well, if they're just mutating and producing cancer, it's going to be really tough to do. And somebody else in the room went, but there are cancers that run in families. So these people are being born with those mutations already present. And that's increasing their chances of getting, there's different types of ones, but the one they suggested was breast cancer. Breast cancer runs in families. And they said, maybe we can use that to figure out what gene is mutating in other people that they're being born with. How do you do that? Well, you go out to a bunch of families where breast cancer runs in the family. And they already knew the location for various reasons, the locations of genes on, like I had up there, a lot of different genes on different chromosomes. They kind of knew where they were already. So you go out and armed with this list of things whose locations you already know, and then you start to ask people in the families. Can you roll your tongue? How about, can are your ears attached? A lot of the stuff we would have looked at if we did this for the regular lab, if we'd had a regular semester. And then they compiled all the data. And what they found is in some families, the people who wound up being diagnosed with breast cancer also were very likely to have had red hair. In other families, the members who had red hair were actually the ones that were not diagnosed with breast cancer. Both of those suggest a linkage. In one case, the red hair linked to an allele, a cancer allele, and the other one, the red hair not linked to the cancer allele. The cancer allele was on the other chromosome of the two homologous chromosomes. But that narrowed the search, and sure enough, they did find the very first cancer gene isolated, BRCA1, breast cancer 1, is actually on the same chromosome with the red hair allele. So that's what you do. In order to try to find the location of one gene, you try to find matching patterns with genes whose locations you already know. Look for the connections that are connected in linkage. Okay. How are genes normally expressed? Here's a cell. It's a beta cell in your pancreas. What does that mean? Well, as the blood comes out of your digestive system, it goes through your pancreas and liver. And your pancreas cells are kind of tasting it as it goes by. The beta cells particularly are tasting it for glucose sugar. And if there's too much glucose sugar flooding into your blood on its way out through your liver, then the cells will go, oh gee, we need to make insulin. Insulin will tell the liver to get a bunch of the sugar out before it's in our general circulation, and our cells go nuts, burning all this energy fuel. So the message goes, hey nucleus, this is where the codes are. We need insulin, and the message goes in. And my question is, so you're gonna get the code, how many are there? How many codes for insulin do you have in that cell? And the answer, people have trouble coming up with it until they hear it, and then it's like, oh yeah, of course. You got an insulin code that you got from mom, you got an insulin code that you got from dad. You have two of them. This is how the codes get used. So the cells are prepared to use the two codes to make proteins. And so that has an effect over just how much protein you make. Now, that's the question of the first part of the question. Genes in a cell are normally expressed as pairs of codes. Well, what if you have not two codes in there, but three, because you got an extra chromosome? Well, now instead of two codes to make your insulin, you got to have three, and you're going to, right off the bat, make more of it than you expected to make. Is that going to mess up your sugar control system? It might, but that's the thing. There's thousands of codes on any given chromosome. And there are going to be situations where making extra is going to be very, very bad. And this is the problem with extra chromosomes. If you were conceived from a sperm with an extra chromosome or an egg cell with an extra chromosome, as you're developing as an embryo, you're using a lot of genes to make muscle cells and brain cells and bone cells and get the nervous system all going and all the stuff that you do. And very, very quickly, you're going to go into that extra chromosome, that triplet, and you're going to make too much of something that's going to kill you. Embryos that are conceived with extra chromosomes die very quickly. Maybe it lives long enough that you run positive on a home pregnancy test. By the time you get to your doctor, he'll run the test and go, "You're not pregnant." I don't know, I guess the test was faulty. Not necessarily. Is that a lot of embryos die pretty quickly, and one of the major contributing factors is extra chromosomes. Well, what about Down syndrome? Extra number 21, one of the smallest chromosomes you have, and you can survive that. Actually, most Down syndrome fetuses don't make it all the way to term, but a lot of them do. But they're not without effects, right? Having those extra codes has an effect on the way your face develops. It has an effect on the way your brain develops. It has an effect over your lifetime on growth. It has an effect, it turns out that they get a very nasty early onset Alzheimer's because one of the major Alzheimer's genes is on the number 21 chromosome. That extra takes a while to have its effect. But again, it's the effect of having too much of something. All of these other chromosomes will kill you really quickly. There are some people who survive to term with an extra 19, but it really messes them up. But you're overproducing all of these. Look at all of these genes on this chromosome, and that's not even all of them. That's a lot of things to have too many of when you're setting up to make the proteins. So an extra chromosome is bad from conception. It isn't necessarily bad if it happens later on. I had a patch on my scalp that was extra chromosome, actually a missing chromosome, where instead of having a red allele and a brown allele covering the red allele, my scalp divided and the two chromosomes carrying the brown alleles, which were supposed to go this way, both went that way, leaving just the red allele behind. So I had a patch on my scalp that had an extra chromosome, 47, that made scalp cells, and one that only had 45 chromosomes, that made scalp cells. But those scalp cells produced red hair because there's no brown to cover it. But they were scalp cells. That far into development, a cell like that isn't using that many genes. And so that may have been the only major gene on that chromosome it was using, and all it did was change the color of my hair. So extra chromosomes later on can produce birthmarks, it can produce patches in your eyes or different color eyes. It can do a lot of things, but it's not really life-threatening the way that getting conceived right at the beginning is bad for extra or missing chromosomes. Having only one code when you expected to have two. It turns out not to be quite as bad, but it is bad. Now the follow-up is the next question. Let's move that up. We're moving from your pancreas into your kidney. But we've got two different people. We have a female and a male. Now your kidney cells don't care. No, they function as kidney cells, male and female. But one of the things your kidney does is it keeps track of your blood's ability to clot. We have a chemical that it works in our plasma that helps our blood to clot. It's called clotting factor. And it's produced in your kidneys when you need it. So you need it. So the cell goes to the nucleus. Give us the code for clotting factor. Same thing over here. And the code goes in, or the message goes in. Okay, I thought I was out of order. I wanted to make sure I hadn't skipped anything. The clotting factor gene is on the X chromosome. Oh yeah, well, I did skip something. Okay. We'll come back to this. Humans and a lot of different living things use male-female combination to do sexual reproduction. It's not the only way you can do it, but it's very, very common, where one of the genders has the job of depositing lots of food for the offspring to get a start with, and the other one, because it isn't making really big, hard-to-move cells that's got a lot of food in them, the other gender produces cells that have to get to those first cells to fertilize them. That's really the main difference between males and females. Whether you make egg cells or sperm cells. That's how you tell the difference between male and female parts of a flower, is what cells are they making. You don't lift the leaf and look for the noogies, they're not there. So I'm going back, we skipped over question 5, I'm sorry. It's tough going back and forth this way. And the question is, so if there's gender, if there's male-female gender, how do you get it, and it's separate. In flowers, they're both male and female. But if they have separate males and separate females, which some plants do, some of you know which ones that do that. But in animals, it's very common. There are animals that are male and female at the same time. But if they're separate, how do you do that? In humans, a lot of you know, we use XX and XY chromosomes. Females get a pair of X chromosomes, males get an X and a Y. Is that this is known as a mismatch. And the matched pair is the female and the mismatched pair is the male. Now in birds, they use a mismatch, but the matched pair is the male and the mismatched pair is the female. Is that what you wind up with is the chromosome that doesn't wind up in the other gender is specialized to produce that gender. In mammals, our group, a whole bunch of genes on this little bitty chromosome are involved in making the embryo become male as the embryo develops through embryo and fetal development. In birds, the mismatched chromosome produces females. The default is male. In mammals, the default is female. But it's a mismatch. Not the only way to do it. There's 3 ways. You can also have it, this happens in a lot of insects, where you either get one sex chromosome, and that's going to be one gender, or 2 sex chromosomes, and that's going to be the other gender. 1 is often the male, and 2 is often the female. And that's called a singlet doublet. And there isn't a specialized gene for it, but overproduction of some proteins causes the development of one gender over the other. Now, there's a lot of animals where the chromosomes don't do it. They're not conceived as one gender or the other. They become one gender or the other reacting to their environment. Alligators, for instance, they bury their eggs in what's essentially a big compost heap. And in the warm spots in the compost heap, those eggs develop male. In the cool spots, they develop female. I think I've got that right. I may have that backwards. But it's temperature dependent. And the aspect of the environment that makes the one gender happen varies a lot from species to species. And it isn't always early. In some fish, especially schooling fish, they may have basically get to the point where they hit puberty. And then, depending on the ratio of males to females around them, if there's more males, they become female. If there's more females, they become male. They can go, that's their environmental cue to become male or female. And in fact, there are some fish where if a big old predator swoops in and takes a whole bunch of the school of fish, if there are mostly males left behind, a bunch of them will just convert to females. And vice versa. If there are a bunch of females left behind, a bunch of them might convert to males. So environmental is kind of fancy. You kind of wonder why it's not more common, because it's certainly much more flexible than this at conception deal. But there's different ways of doing it. We needed that before I went on to the question I was on before. That's why I got halfway to the question and went, wait a minute, we should have covered this already. The gene for clotting factor is on the X chromosome. So some of you watching the video have 2 of those. I only got 1, right? I have a Y. I don't have 2 pairs. I don't have 2 X's. I don't have a pair. I've got the mismatch. So I can only produce 1 code, whereas you can generate from 2 codes. So right off the bat, you can crank out twice as much of this as I can. Now, is that a big deal? Depends on what the gene is, right? Overproduction, underproduction isn't always going to be a factor with a whole bunch of things that you make, but this could be a problem because that X chromosome is one of the biggest chromosomes we have. It's got thousands and thousands and thousands of genes on it. And this idea that everything I need from the X, I only got 1 code for, and the females have 2 codes for, is potentially a major problem. That imbalance is potentially a major problem. The X is not a female chromosome. It just has a whole bunch of regular old genes on it, including clotting factor. So the question is, is there some compensation system to keep this from being a major problem? And it turns out there's a couple of ways to get around this problem. If we were fruit flies also have XX and XY female-male genders. But what a fruit fly does is that if you need an X code, it automatically makes a copy before it comes out to be produced into protein. So you get 2 codes. They're the same code, but you're getting double before it ever comes out to where you're going to use it. So both genders, from everything on the X chromosome, they make 2 codes and it goes. We don't do that. If we can't make 2 equal to 2, we make 1 equal to 1. And what happens in a female cell is there's a functional X chromosome, and then there's a chromosome that's all skewed up really, really tightly, so that almost none of the genes are usable. And so if you're female and you need clotting factor, you go, give me the code, only one of them's gonna come out. Now the difference there is that which one is shut off is kind of flipping a coin random. So if you have 2 different alleles for clotting factor, then half of your kidney cells are making from one allele and half of them are making from the other allele. That turns out to be important for some things we'll talk about in a couple of minutes. But some animals can make 2 equal to 2, we make 1 equal to 1. So that my expression of my X genes matches a female's expression of her X genes, and neither one of them is producing a superhero. Sorry, some of you understand that reference. There are some animals that don't even worry about this, is that they've managed to produce a sex chromosome that doesn't have any genes on it that over or under production really is an issue with. So it does turn out there's another workaround you see kind of rarely in the animal kingdom. Usually it's either doubling or shutting one off. Speaking of, what's a bar body? Bar body is that little wrapped up X chromosome that makes the males and the females equal in expression of X genes. Is that you need that one shut off pretty much to have this expression equal to that expression on all the many genes that are on the X chromosome. Named after Dr. Barr from Long Island, I think, New York. So this brings us to, oh goodness, this is going really long. Sorry about this. I try not to go over half an hour, but I've got to, because I only got a couple of color blindness is a sex-linked trait linked to the sex chromosomes. And most of you know that males are more commonly colorblind than females, to which the question is, so if it's linked to a sex chromosome, which one is it linked to? And I'll tell you right now, don't fall into the trap. If it was on the Y chromosome, then I'd be able to see color, and women wouldn't be able to see color because they don't have a Y chromosome. So it can't be on the Y. The Y is just about making things male. Color blindness isn't about male. It's on the X. Here's how that works. Here's a male and a female, not in that order, both of them carrying a colorblind allele, which messes up their ability to, their color receptors in their eyes, usually the red-green ones. Well, actually, in this one, it is the red-green ones. She's got a colorblind allele. She's got another X with a good working version of that protein. She can cover this up. In women, color blindness is recessive. In men, there's no gene over here to do anything, so dominant recessive doesn't make any sense. There's only one copy. So if you get the colorblind allele, ta-da, you're colorblind. You can be a colorblind woman, but you need to get 2 of these. And they're not that common in our gene pool, so the odds of getting 2 of them together are pretty unusual. You don't get a lot of red-green colorblind women. It does happen. So that's the explanation for why it shows up more in men than in women. Equal numbers of men and women are gonna have one colorblind allele, but the women are not gonna be colorblind when they have it. The men are, because they got no balancer, they got nothing to cover it up. So now we're gonna become marriage counselors. Woman comes to you and is worried about having kids with this colorblind guy. She knows you took a genetics class, so she wants you to tell her what the risks are. And so the first thing you say to her is, so you're not colorblind, right? She goes, no. I said, how about your dad? No. Any of your uncles? No. Your brothers? No. Grandfathers? No. Because it's going to show up in the men in the family more often, right? So you're trying to figure out if there's any colorblind alleles in the family. She could be hiding one. But it kind of looks like she isn't. So that's why I crossed the CB off on this X chromosome. She's got 2 good alleles. But we know he's colorblind. He's got the colorblind allele. And then she said, well, so what's going to happen with our kids? And you're going, well, let me figure it out. And think about this. What are the odds that their sons are going to be colorblind? What are the odds that their daughters are going to be colorblind? What are the odds that their daughters are going to be carriers? This messes with people's heads. I used to have people do this on their own. They would mess it up. I don't make them do that anymore. Here's the thing everybody forgets. To make sons, dad needs to send that on. So the only place the sons are going to get their X is from the mom, and she's got 2 good alleles. So the sons can't be colorblind and be sons. The daughters will get dad's colorblind allele, but they'll also get mom's good allele. So they're not going to be colorblind either because it's going to get covered up. It's going to be recessive and covered, but they're always going to be carriers. So really, she should be worried about her grandkids, because this daughter has a 50-50 chance of producing colorblind sons, because half the time the sons are going to get this X, half the times the sons are going to get that X. But the sons and the daughters are fine. They're not going to be colorblind. Okay, on to the last question, which I'm going to go quickly through. We talked about how it's really bad to get extra chromosomes, and how the number 21 is about the only one that you're okay with. Well, it turns out people get extra X's and Y's too, sometimes bunches of them, but they don't have the same sort of horrible effects that getting an extra number 5 or an extra number 9 or an extra number 12 does. And your last question is to explain, what is it about getting extra X's that is not going to really make it bad for you? And what is it about extra Y's? It's a different answer. We have talked about it. It has come up as we went through this part, but you need to figure it out. And that's your last question. And that got me to, make this not super, super, super long.